Hello guys, welcome to the Parson series, and this is part 1 of your chapter number 4. So in this chapter, it is basically concerning the neurology of the vision. I think it's a very important aspect because we always fear of neurophthalm. So this will be, like basics of the neuroophthalmology. So let's talk about the visual pathway. Visual pathways have some unique features which are better understood if the afferent tracts of the common sensation are compared with the vision. So let's start with the afferent tracts of the sensation. The sensory impulse of common sensation in a limb. Now they are actually trying to compare this same pathway in a limb, and then we will see the similarities what are happening in cases of our eye. So the sensory impulse of a common sensation in a limb is carried by a nerve fiber along the sensory nerve and the dorsal spinal root of the cord. Then it travels up in the posterior column of the cord to the nucleus gracilis or the nucleus cuneatus, as the case may be, and this entire course is along the processes of a single cell or the neuron of the first order. So this actually you know better than me that this is the whole pathway, how the sensations from a limb are carried through the nucleus gracilis or the nucleus cuneatus, and how they are graded as the neurons of the first order. Now this impulse is taken up by your nucleus gracilis or the cuneatus by a second cell. Okay, and then it is carried along the nucleothalamic tract or the medial lemniscus to the opposite thalamus, and these will contain the neurons of the second order. Right, now a third cell which is situated in the thalamus, neuron of the third order, that is carrying the impulse, and then finally it is transformed into sensory perception. So whenever we have to convert a impulse into a sensory perception, we have to run through the neurons of first order, second order and the third order, like so it is. So the same thing will happen with the anatomy and the physiology of the visual pathway also. So that is why they have taken a simplest of example, and now we'll be comparing the same thing in the eye. So what they are saying, the pathway of the visual afferents is very unique, and the end organ that is lying in the neural epithelium is very basic, that is the photoreceptors, the rods and cones. And in this way only, the photoreceptors are very unique. They are very unique in the sense that these photoreceptors, right, at every place we have got only the sensory end organs. But in cases of eye, in these photoreceptors, that is your rods and cones, we have the generation of the impulse. And because these are the only photoreceptors are the only receptors where you have the generation of the impulse, they are considered to be the first order neurons. There is a lot of controversy that whether these are first order neurons or the bipolar cells are the first order neurons. So yes, in that way they are just the receptors. But if you consider that first order neurons are the neurons where you have the generation of the potential, so eye is the only organ where you have the generation of the potential, right? The first conducting nerve cell or the neuron of the first order is the bipolar cells of the inner nuclear layer of the retina. Now, though, there are certain school of thoughts which is differing. Certain books say that it is the rods and cones which are the first order neurons. Certain books say that it is the bipolar cells which are the first order neurons. So here they are saying that the neurons of first order is the bipolar cells. But again, this is questionable because I'm going with the latest edition of Yanov, which says that rods and cones are the first order neurons, and we can verify it also because the first point of the potential generation, the first point of the potential generation is your rods and cones. So if you consider it in that manner, then your first order is rods and cones, second order is your bipolar cells, and bipolar cells will be present in the inner nuclear layer of the retina, right? Now with its axons, so axons will be lying where? In the next layer, that is in a reticular layer. This microscopic cell corresponds morphologically to a dorsal root ganglionic cell, which in some cases have long processes from the tip to the toe, that is of the spinal cord, right? Now, if you consider because they are considering the neurons of bipolar cells to be first order, so they are saying the ganglionic cells to be second order, but if we consider the photoreceptors as first order, they will become the third order neurons, the processes of which pass into the nerve fiber layer, and then they are passing through the optic nerve and finally to the lateral geniculate body. So order will remain the same. We will be starting from our rods and the cones, then you are going to the bipolar cells, then we have got the ganglionic cell, and then we have got the lateral geniculate body. So in this way we have got the fourth order neurons going up. Here a new cell, that is your neuron of the third order, takes up the transmission of the impulse, and they are traveling by the way of the optic radiations, and they are going to the cortex of the occipital lobe. So this is the so-called visual center, right? Now the morphological identity of the two systems is apparent despite the great anatomical differences. You know, if you see anatomically, obviously there are a lot of differences, but morphologically both of them are same, and the peripheral optic nerve proper corresponds to a bipolar cell in the inner nuclear layer and the inner plexiform layers of the retina. So you already know the structure of the optic nerve as a part of central nervous system, and if I consider the homologous, they are homologous with the medial lemniscus in the medulla and the pons. So we are trying to explain you all these things by explaining you the homolog organs which are lying in the central nervous system. Now coming to the retina, optic nerve and the chiasma. The course of the fibers from the various parts of the retina, we are seeing it in the picture also, we will go to that. In general, it may be said that the fibers from the peripheral parts, they enter the periphery of the optic nerve. So first they are entering the periphery of the optic nerve, while those parts of the retina which are near the optic disc, they enter the central area of the nerve. Now we have got two kind of fibers which are entering into the optic nerve. One is your peripheral fibers, one is your central fibers. So what they are saying, that the fibers which are coming from the peripheral parts, they will be going into the periphery of optic nerve, same, and the fibers which are going from the parts of retina which are near the optic disc. So near the optic disc means the central part of the retina. So I can say that those fibers which are in the periphery of the retina, they are going in the periphery of the optic nerve, and those parts which are from the center of the retina, that is near the optic disc, they are going in the center of the optic nerve. They maintain this relative position as far as the chiasma is concerned. Now fibers from the macular region, if you consider the fibers which are coming from the macular region, they are behaving differently. They enter the nerve on its outer aspect, whereas they are spread over an area which is triangular in section. So those fibers which are coming from the macular area, they are spreading, okay, and in the area in which they are spreading, they are triangular in section, with the apex, if you consider the apex is towards the center of the nerve. So this is actually triangular in the section, and this apex is towards the center of the nerve, okay. Now these fibers are your papillomacular fibers. Okay, so papillomacular fibers, papillo means the optic disc and macula means the macula. So those fibers which are actually connecting the optic disc and the macula, these are more centrally situated, so that they are in the posterior part of the nerve. They are all in the center. Okay, so if you go towards the posterior side, so these macular fibers are lying in the center there. They are triangular in section. All these things are important. Now tracing the nerve fibers, if you go still further backwards, a partial decussation occurs in which the nasal fibers cross in the chiasma, whereas the temporal ones enter the optic tract. So this is the same thing that you already know, that the nasal fibers are decussating in the optic chiasma while the temporal fibers are descending on the same site, right? The axons of their corresponding neurons. Okay, if I consider the axons of corresponding neurons, that is, they are going into the third order. Now from our side, our third order is actually the ganglionic cells. From their side, their third order is lateral geniculate body. So they are saying that they are going and they are synapsing in the third order, that is your lateral geniculate body, and then they are going with the help of this after this lateral geniculate body, they are going to their what you called as optic radiations, and finally they will be going the visual cortex. So they have just theoretically told you whole of the part of your visual pathway. All right. So this visual cortex is actually like at the tip of the occipital pole, right? So each half of the macula will be representing the corresponding part of the occipital pole. So actually what happens, the tip of the visual cortex represents the macula, and that is what we always say, that the tip is having extra blood supply, that is your middle cerebral artery. Whole of the visual cortex has just one blood supply, that is posterior cerebral artery, while the tip has extra blood supply, that is your posterior cerebral artery. And that is why whenever we have a problem with the posterior cerebral artery, this middle cerebral artery continues to supply this tip of the visual cortex, and that is why we have the macular sparing. So this is the same picture that you keep on seeing of the visual pathway. We have got this your optic nerve is there, then the chiasma, it is crossing on the other side, then we have got the foveas. Okay, now this portion is your optic chiasma, then we have got this optic tract, then we have the lateral geniculate body, then you have the optic radiations, and finally the visual cortex. So this you already know. Okay, now coming to the other things. Now what they are saying, similarly the fibers from the peripheral regions of the retina, they form two distinct groups, that is your temporal half and the nasal half of the retina. The distinction is exact, as if a vertical line divides the retina into two halves at the level of the fovea. Now what is happening, we can divide this into nasal half and the temporal half, and from where this line is actually passing. So this line is actually passing through the retina, and it is dividing the retina into two halves, and by passing through the foveal region. Fibers from the temporal half of the retina enter the chiasma, and they will be passing through the same side, this we already know, and then they are going through the lateral geniculate body. But if you see the fibers from the nasal half of the retina, from the nasal half of the retina, they are going through the chiasma, they are decussating, and they are passing into the optic tract of the opposite side. Now the arrangement being such that the direct and cross fibers of the alternating lamina, they are ultimately going into the lateral geniculate body, and from this lateral geniculate body they are going to the optic radiations, and finally to the occipital lobe, that is the visual cortex. So this you already know. Okay, now projecting this outward, such a lesion will also cause. Now what they are saying, that if it follows that a lesion of one occipital lobe or the optic tract will cause the blindness of temporal half of the retina of the same side and nasal half of the retina on the opposite side. So basically they are talking about the homonymous. They are talking about the homonymous hemianopia. Homonymous kind of hemianopia, that if we are having a lesion in the optic tract, or after that you will have a problem in the visual field of temporal side, temporal fibers of the same side and the nasal fibers of the opposite side, because this is the distribution like. So because you know you are having the temporal half of one side and the nasal half of one side, so obviously you will have the same side of both the fields going. So that is called as homonymous hemianopia, right? So see, so projecting this outward, such a lesion will cause the loss of vision in the opposite half of the binocular field of vision, and we are having just half of the fields which are blind. That is why it is hemianopia. And if you have same half of the fields, then it is homonymous hemianopia. If you have different half of the fields, like I can have bitemporal also, I can have binasal also. So if I have that kind, then it is called as the heteronymous kind of hemianopia. The afferent pupilloconstrictor fibers have a similar semidecussation. Then we are talking about the optic radiations. Optic radiations the visual fibers in the optic radiations, they run behind the motor fibers in the internal capsule. Now this, if you remember, I always tell you in the classes also that whenever we have some stroke or some cerebrovascular accident, mostly the motor fibers are affected in the internal capsule, and we also have the sensory fibers which are passing behind these motor fibers in the internal capsule, and thereafter they are separating. So if I have some problem, some cerebrovascular accident in the internal capsule, then along with the stroke, the person will also have a problem with the visual fields. Thereafter they are separating, and the ventral fibers running forwards into the temporal lobe, before they turn backwards to the lower portion. So they want to tell you that how these fibers are actually related with the internal capsule. That's it. Then we come to the occipital cortex. Now the occipital cortex, you know, in and about the calcareous fissure differs from the cortex elsewhere in the position of a white line. This is called as the line of Guarnieri, and this area, you know, which is the primary visual area or the visual sensory area, this is the cortical projection of the corresponding halves. The same spatial arrangement is maintained in this projection. The parts above and below the calcareous fissure. So what they are saying, that this occipital cortex, the area of the occipital cortex which is near your calcareous fissure, that differs from the cortex, from rest of the cortex. So that area, the part which is above and below this calcareous fissure, will represent the upper and lower part of the corresponding quadrants of both the retina, and the posterior part of the occipital lobe will actually represent the macula. So if this is the calcareous fissure, then the above area and the lower area, this will represent the corresponding field. And if I talk about the posterior part of the occipital lobe, then this will actually represent the macula, right? So this is the representation in the diagrams which they have given you. Now if we come to the neurology of the brain in relation to the vision, unraveling the mysterious functioning of the living brain has been tantalizing but elusive quest for imaginative researchers over centuries. So here we try to read about the radio and of the integration there. So how we can correlate the visual system with the neurology of the brain, and neurology of the brain can be studied with the help of slices in the MRI and the CT scan. Okay, so what they are saying, that meticulous recording of the neurological examination performed on the people who had suffered from brain injury or stroke have subsequently recovered, and also the observation noted during the surgery. So new options have come, because earlier we were not that much capable of reading the slices of the brain and compare the neurology of the brain with the vision, but now we have. So we have got better things available. We have got MRI, that is called as the functional MRI. Even the positron emission tomography, that is PET scanning, is also there. And now we are able to see the working of the brain in vivo too. So PET scanning, what is the basic principle? You must be knowing this better. PET scanning uses the radioactive labeling of the substances. Just say we have glucose administered to the subject prior to the scan. So this glucose will be utilized by the brain. So what is happening? The brain will be metabolizing this radioactive labeled glucose which remains within the cells, releasing the positively charged particles called as the positrons. So what will happen when brain will metabolize this glucose which is radioactive labeled, then it will release the positively charged particles or the electrons. These are called as the positrons. Gamma radiations are released by the collisions. Now what happens? There is collisions of these positrons with the neighboring electrons. So these positrons which are emitted, they will be actually going on collision with the electrons, and they will release what you called as gamma radiation. So finally we have the release of gamma radiations there. Now whatever are the radiations which are released, they can be captured. Color images coded for different values of the map, the parts of the brain which are more active, and hence they are using more glucose, they can be captured now. This technique can be used to show which sites in the brain. So what will happen? Now I can get a site that where it was actively metabolized. So I will know the sites in the brain which are activated in the pathological conditions, and also during the normal physiological process like thinking, expressing emotions, seeing and reading. So you will get to know which parts of the brain are actively utilized during the pathological conditions. Okay, a brief description is presented here of the complex coordination by various parts of the brain involved in interpreting the visual environment. Now, primary visual cortex. If I talk about the primary visual cortex, then this demonstrates a peak of activity in the PET image following the presentation with a bright flashlight. So, if a less intense stimulus such as a word is presented, okay, then the PET evoked image is less distinct, and this will be harder to interpret also if you are using a less intense image. But if you are getting the high intense image, the response involves the changes in the visual receptors which registers the stimulus and also possibly gets transmitted to several other sites which are concerned with the language, memory, learning, emotion, depending on what the word is presented. Okay, now if I talk about the English speaking, so English speaking literate subjects were presented first with an array of arbitrary symbols, then with real letters grouped in units which had the average length of words, but they were unpronounceable. Okay, next letters arranged to appear like normal words which could be pronounced but were not recognizable words, and finally genuine English words were presented. So they did, you can say an experiment was done to with the people who were English speaking. So they were given three set of words. First, those words which were unpronounceable, these words were given. Next, the words which were pronounceable but they were not recognizable. This was the second group. And third, they were given the genuine English words. So the PET, and we recorded the PET activity. It clearly showed that there was actually sharp increase in the activity in the area known as the medial extrastriate cortex in the left hemisphere when the subjects applied the third and fourth level of the task. So whenever the subjects are given more complex tasks, the third and fourth level of tasks, then we were getting a sharp increase in this activity, which is called as the extrastriate area of the cortex. Subjects with brain injuries affecting this site have been known to have difficult in reading the words, and often despite having no problem with speaking and writing. So what they are saying, that those people who have a brain injury, now try to understand, if a person has a brain injury, he will have a problem in reading the words but no problem in speaking and writing. Reading the words is giving a problem, so that is related with the vision there. Okay. Similarly, the utilization of the technique of functional MRI. Then we can also utilize the functional MRIs for engaging what is considered to be a simple task of reading. Different parts of the brain, attention, vision, memory, they must be activated and coordinated. So if you want to see the things by the functional MRI, okay, that has been shown for engaging what is considered as. So what are the things that you need to do if you want to see these things in the functional MRI? The brain must focus on the task of reading, so that paying attention involves the frontal cortex and the thalamus. So first we are involving the frontal cortex and the thalamus, then the words must be visualized, because when you are visualizing the words, we are engaging now the occipital visual cortex. Then we have to make the sense also, whatever words you have seen, in order to make sense, okay, of the seen words, the visual information will go to an association area. In this association area, you will see the symbols or the letters visualized and you will get the meaning of that word. This knowledge is of importance in the understanding of physiological basis. Why this is important? Because not only we are saying that integration is important, it is actually taking place. So whenever we are dealing with the dyslexia, it has been seen that there is a cortical atrophy or the hypoxic damage in the children who cannot see, because those areas which are not being used, okay, because there is no stimulus to those areas, so there becomes these areas become atrophic, and it has been seen that those children who cannot see, there is a atrophy of this area, okay, though their distinct afferent visual pathway may be intact, and this had certain implications in the teaching and learning as well. So this was an important aspect where we tried to compare the neurology of the brain with the limbic system also. We tried to see how complex it is, how actually the brain and vision are interrelated. I think this is a beautiful concept and could be studied with the help of radiological techniques, that is your functional MRI and the PET scan. So I think this was all for this part. In the next part, we'll be continuing with the pupillary pathways and reactions. Thank you and happy ophthalmology.